Hello guys, today we are going to discuss about monohybrid and dihybrid cross. Okay, in the monohybrid cross, if you have any doubts regarding the terminology that have been used in this video, you can refer to the previous video which I have done, that is basic terminologies used in genetics. Okay, here we have taken two diversely homozygous parents. Okay, the gametes will be capital T and small t, and in the F1 generation this will be capital T, small t. How this has came, now I am simply explaining you. Capital T from here, small t from here, this capital T from here, this small t from here, this capital T from here, this small t from here, this capital T from here, this small t from here, right? This is the F1 generation. This is what we got in the F1 generation. And again, if we do crossing between these two F2 generation, F1 generation, we'll get the F2 generation. So we'll write the gametes of these two. It will be capital T small t and capital T small t, right? And in the F2 generation, we will get capital T, capital T small t, capital T small t, small t. How this has came, just I will explain you. This capital T from here and this capital T from here, this capital T here, this small t here, this capital T here, this small t here, this small t and this small t. Like this it has came, and this generation have been formed, right? Like if we observe in this generation, there are three plants with capital T. Capital T resembles tall plants. So if we see phenotypically, the three plants are tall and one is dwarf, because it doesn't have any dominant allele in this, which is expressing. Here we are having only two small t's, so it will be dwarf. And here we are having at least one capital T in each case, so it will be tall. So the ratio of F2 phenotypically will be 3:1 is the phenotypic ratio. Phenotypic means physical appearance. Like if you see the plants, you can say three plants are tall and one is dwarf, right? And the genotypic ratio will be, see here the genotypic. Now we have to come to the genes. Genotypic means gene level. So here we can see two are having, like one plant is having both capital T and capital T small t in two plants, small t in one plant. So this will be 1. These two are similar, so we can write 2, and this is again 1. So the genotypic ratio will be 1:2:1, right? This is the thing. And now I want to explain you about Punnett square. There is a scientist named Reginald Punnett. He discovered this Punnett square. Let's understand what this Punnett square is actually. It's normally, for example, like this we have to take. One side we have to take the female and another side we have to take the male parent, right? For example, if the female parent is small t, if the male parent is capital T. here these two, see, just try to understand this one and this one will combinedly together form as capital T small t. Now this one and this one will form as capital T small t. This one and this one will form as capital T small t. This one and this one will form as capital T small t. Right? The same thing now we are doing in normal method. Now we are taking this male parents capital T into this female parents small t, right? The gametes from this capital T will be capital T, and here the gametes will be small t. So the F1 will be capital T small t, capital T small t, capital T small t, capital T small t. This will be the F1 generation. In the F1 we all got this capital T small t, and in the Punnett square also we are getting the same. This will be somewhat difficult if we go in case of dihybrid and trihybrid crosses. So Punnett square is easy to make our work easy. So this has been done. Yeah. Now the phenotypic, here all plants are capital T and small t. So all is having one capital T. So phenotypically if we observe, like normally if we observe, all plants will be tall. So we can say phenotypically all are tall, and genotypically are also all are equal. So we can say all are heterozygous, right? If you have any doubt regarding heterozygous, just I will explain you that also. If capital T or capital T small t, if this case is present, this case is called homozygous, and if any two different alleles are present, then this is called as heterozygous. Now we will do the Punnett square for this F1, capital T small t. I will take Punnett square. Let's take this is male, and again female parent capital T small t, capital T, capital T small t, capital T small t, small t. Just understand how this came. This small t corresponding to this small t will form as small t. This capital T corresponding to this small t will form as capital T and small t, and this capital T corresponding to this capital T will form as capital T. This capital T with this small t will form as capital T small t. Just the corresponding rows or columns, just you have to observe and fill the table. You have understood, right? Now the phenotypic ratio, see, all these three plants are having at least one capital T. So we can say these are all tall, and this one will be dwarf, right? And the genotypic ratio will be, this one is having capital T, 2 capital T small t into 2 small t. So it will be ratio 1:2:1. This is the same ratio what we have done here, like this type of crossing. So it will get the same results as in the Punnett square also. Now let's go into the dihybrid cross. If you take any two characters into the consideration, so it will be called as dihybrid cross. Now we can say round and wrinkled seeds we have taken in one set, and in another set yellow and green seeds we have taken, right? So we'll represent this round and wrinkle as capital R for round, for wrinkle small r, for yellow capital Y, for green small y, right? The P1 generation will be capital R, capital R, capital Y into small r, small y. For easy understanding, I have taken here homozygous traits, right? And the gametes which will be formed from this is capital R, capital Y, capital R, capital Y. From here it will be small r, small y, small r, small y, right? The F1 generation will be capital R, same. Capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. So this one will be the F1 generation. Let's take a moment to understand this. If we do like, if we got this one, see, all the F1 will be similar in the dihybrid cross also, same as the monohybrid cross. So if the gametes, like if we write gametes for these, we'll get capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, right? And for again any other parent also will take, we'll get the same gametes, right? These are the gametes that we are getting, right? If we cross between these two parents, let's understand this in the Punnett square, so it will be easy. Now if we cross between this F1 generation, so the gametes will be capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. Let it be the male parent, let it be the female parent. Now we are crossing between these two parents, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, right? Let's draw some rows and columns just to avoid the confusion. Right, here the gametes will be capital R, capital R, capital Y, capital Y, capital R, capital R. See, here capital R is there, corresponding capital R I have written, here capital R, here capital R, here capital Y is there, I have written capital Y, here small y is there, so I have written small y, right? Right, now just observe how many round and yellow seeds are there. If we have minimum one capital R and capital Y, so we will get round and yellow seeds. If we have one capital R and both small y, then we'll get round and green seeds. If you have both are capital R and smaller and one capital Y, here we will get wrinkled and yellow seeds. And if we have smaller and both are small y, here we will get wrinkled and green seeds. Right here, till now here we have understood, you guys have understood, I guess. So let's go inside this. Now if you have any one capital R and capital Y, here we will get rounded yellow seeds. Now we'll circle this with orange color. Right, one capital R capital Y, so it is there. Capital R capital Y is present in this. Capital R capital Y, capital R capital Y is present in this. See, capital R capital Y is present in this. Capital R small y, no, this won't be happening. Capital R capital Y is present in this, so we can circle this. Here it's not present. In this we can see capital R capital Y, we can circle. Capital R capital Y, we can circle. These are small. Again you can see here. See the orange. Now let's count number of orange boxes. 1 2 3 4 5 6 7 8 and 9. So these are 9. Now let's circle these two. Capital R is there, small y is there. Okay, now we are round and green, we are circling with this with pink. Here we are having one capital R capital Y, and again here we are capital R small y. Here also we are having one capital R small y, and again here you can observe, here we are having one capital R and small y. So this will be round and green. Now let's take another color, green. Here in the third case we are talking about, here we have to have both small and at least one capital Y. So here both small r's, at least one capital Y. This one we will select. Both smaller, at least one capital Y. Here we will select. Both smaller, at least one capital Y. Here this we will select. So this also is in the ratio. Pink is in the ratio 1, 2, 3. So we'll write here 3. And round and yellow. These 3. 1, 2, 3. This is also 3. Smaller, small by small y. And this is only 1. So here we will write it as 1. So the dihybrid ratio will be 9:3:3:1. This is our dihybrid ratio, right? In this case 9:3:3:1. So this is our normal dihybrid ratio. If you have any doubts, just ask me in the comment section. Thank you.